Now that we know all about the light-dependent reactions of photosynthesis, let us look more closely at the light-independent reactions, or also known as the dark reactions. So the light-independent reactions, or the dark reactions, would also be the portion of photosynthesis where carbon dioxide is reduced to become sugar. And the power of reduction here is provided by the NADPH that was produced during the light reactions. And of course, the energy is from the ATP, which is also produced during the light reactions. So how is carbohydrate synthesized from carbon dioxide? Okay, so this process of carbohydrate synthesis, when studied by scientists, somehow allowed us to classify plants into three types, depending on how they process the carbon dioxide to become sugar. So scientists were actually able to track the carbon in carbon dioxide into how it's incorporated in a 3-carbon intermediate in plants we call as C3 plants. Okay, so here we can see carbon in carbon dioxide incorporated in 3-phosphoglycerate. But in reality, the carbon dioxide is actually not incorporated directly into the 3-phosphoglycerate. It is first incorporated into a 5-carbon compound called ribulose biphosphate. So as we see here, this is the intermediate. The intermediate now has 6 carbons instead of 5 because 1 carbon dioxide molecule has been incorporated. And this intermediate is unstable and its tendency is to split into 2 3-carbon compounds, and that's the 3-phosphoglycerate that we see here above. Alright, so these plants where scientists were able to track carbon and carbon dioxide to be incorporated into 3-phosphoglycerate are known as C3 plants. And most of the plants that we are aware of are actually C3 plants. But you will learn later on that there are actually plants as well that are very common in our environment that are not C3 plants. So how is carbon dioxide incorporated into ribulose biphosphate? So we see in this process of carbohydrate synthesis, which is also known as the Calvin-Benson cycle, or the fixing of carbon dioxide, we see here an enzyme called Rubisco. Rubisco stands for ribulose biphosphate carboxylase oxygenase. That's because this enzyme has 2 tendencies. It can either work with the carbon dioxide, which is the normal thing for plants, or it can incorporate oxygen, which is a little bit detrimental to the process of carbohydrate synthesis. So, as early as now, I think it's good for us to recognize that although Rubisco does its work in the carbohydrate synthesis, there is a tendency for it to do the wrong thing, to catalyze the wrong thing. Alright, so as of the moment, let's talk about what it does right. So here we can see 6 carbon dioxide molecules incorporated to 6 ribulose biphosphates to form a 6-carbon compound, 6 carboxylases. Okay, and these are the intermediates. Now these carboxylases are unstable and they will tend to split, so forming 12 phosphoglycerates as we see here. Now, these 12 phosphoglycerates are then phosphorylated using the ATP produced from the light reactions. And once they are phosphorylated, they now have two phosphate groups, as you can see here. So they are now known as biphosphoglycerates. And one of these phosphate groups, the one highlighted in yellow, is going to be reduced with the help of NADPH, which was also produced during the light reaction, and as you can see here, replaced by hydrogen. So this is the process where the reduction is actually needed, the reduction power of NADPH. So you now have 12 GAPs, okay, or glyceraldehyde phosphates. And these GAPs are going to be incorporated together to form fructose 1,6-biphosphate. And then turned into its isomer, okay, glucose. And may also be combined with existing glucose already to form sucrose. So as we know, the table sugar that we have right now, sucrose, is actually from a plant, the sugar cane. Okay? And the sugar cane produces a lot of sucrose through this process. Okay, this carbon fixation process. So it is here that the final output of photosynthesis, the food for the plant, the sugar, exits, is created. But it's not all of the glyceraldehyde phosphates that are made into this sucrose that we have here. Most of them actually go back into the cycle or are reused into the cycle. So they are phosphorylated again here by ATP. And the 10 GAPs, which contain 3 carbons each, will become combined to form 6 ribulose biphosphates again, which will be the substrate again for Rubisco, the enzyme. And when Rubisco does its work correctly, it will incorporate 6 carbon dioxides with the ribulose biphosphate. So this is the carbohydrate synthesis. And the output would be the sugar that we see here. And of course, the sugar will be stored in the plant for long-term storage and becomes starch. All right, so let's talk about that other inefficient pathway where Rubisco is involved. So shown here in blue arrow would be the correct or the ideal pathway for carbon fixation, where carbon dioxide with the help of Rubisco is incorporated in ribulose biphosphate to form carboxylase intermediate, and then that will split into PGAs. But the other pathway, which is the non-favorable one, as the name of Rubisco suggests, ribulose biphosphate carboxylase oxygenase, the oxygenase portion, is the portion where Rubisco would wrongly incorporate instead oxygen gas into our ribulose biphosphate to form oxygenase intermediate, but only one PGA. And then there's phosphoglycolate, as we see here, and 2 hydrogen ions. And these 2 are waste products. They are literally useless. And for the plant to get rid of them, they have to pass through a different pathway, as we see here. Pass through chemical reactions, be converted into glycine, and eventually processed in the mitochondrion and expelled from the plant as carbon dioxide, a waste product. So this process is known as phosphorespiration or photorespiration. because it releases carbon dioxide in the same manner as cellular respiration does. So certain adaptations have been employed by certain types of plants to avoid that inefficiency of rubisco, that oxygenase part. And this results into the C4 and the CAM plants. Examples of C4 plants are the grasses, like sugarcane, for example, tropical grasses. Then examples of CAM plants are those plants that are succulents, and they usually are found in dry, hot environments. The reason why these plants would need to adapt is because their stomata, okay, would need to close early on every day. Okay, every day their stomata would need to close because they live in a warm environment to prevent the excess evaporation of water. So when their stomata close, the chance of letting carbon dioxide enter would now be very, very low. And oxygen gas might compete with carbon dioxide as a substrate for rubisco. So what these plants do is, as you can see in this illustration, they locate their carbon fixation deep within the leaf, in the bundle sheath cells of the leaf. So what they do is they let carbon dioxide in through their stomata, and the carbon dioxide is actually incorporated into phosphoenol pyruvate, and that's converted into oxaloacetate, and then that furthermore becomes malate or malic acid, and it's the malic acid that is transported deep into the bundle sheath cell, where it gives up the carbon dioxide that was attached to it, and that's the only time that rubisco is able to use the carbon dioxide to do the carbon fixation, as we see here. So this reduces the tendency of rubisco from attracting or from working with oxygen gas, because carbon dioxide is chaperoned into the bundle sheath cell using oxaloacetate and then malate. For C4 plants, the reason why they're known as C4 because oxaloacetate is actually a 4-carbon intermediate, as we see here. So they're known as C4 plants. And for CAM plants, the name actually comes from crassulacean acid plants, okay, CAM plants, and that crassulacean acid is actually this malate here, or malic acid. So the cacti actually produce malic acid, and at night, that's the one that delivers carbon dioxide into its rubisco enzyme. So malic acid is sour, so if you try and taste a leaf of a cacti early in the morning or at night, late at night, it will actually taste sour because of this crassulacean acid or malic acid.